I'm Nazar. I'm a postdoc in Civils group. So I started in spring, and so today I'm going to talk about some additional techniques that I'm using along SAXS. And so I'll introduce them, talk about their relevance, and show some initial applications that I started using in my projects. Still lagging.
You have to just be patient. It's so weird. It's having trouble with Windows.
Okay, this is my second slide. Okay, so central in our work is characterization of protein solution, right? So, regarding of our research objectives, we want to know as much as possible about its state. So whether it's some structural changes due to conditions or some type of interactions. So to do that, there are, of course, small angle X-ray scattering that gives us understanding about overall shape and size, kind of like without using any computational tools, we can more or less precise get overall shape and size. Next is going to be a circular dichroism. So this one is good to know about protein folding and overall composition of secondary structures of our protein. And if we want to know what state of our protein solution, like if it's has different oligomers or it's monomeric, how those particles are distributed in solution. Another approach is dynamic light scattering. And finally, the surface plasmon resonance can show us how those molecules interact with each other, how we what kinetics of those interactions. So basically, these are outlines of my talk today, and I'm just trying to show some data that I obtained for some of those experiments and show how we can combine those techniques. So in our work with SAXS, we obtain nice SAXS profiles, and we want to know, like, first of all, we want to know what our molecular weight, right? What radius of gyration, like if it corresponds to what we expect to see. So another thing to look at, if we have our protein folded correctly, right? If we have all secondary structures present in that sample. So for that, we can use circular dichroism. So basically, circular dichroism is another spectroscopic technique. And here I tried to introduce some of idea how it works. So we have light source. It's that light beam goes through monochromator. We are getting linear polarized light. Then some special part of circular polarimeter, which is called photoelastic modulator, gives us two types of circular polarized lights. So right and left circular polarized lights are coming from this modulator. And in our sample, where we have chiral molecules, which are, of course, our proteins, DNA, other nucleic acids, they can absorb those two types of lights differently. And then we are getting different amplitudes of absorption of that right and left circular polarized light. And if we add them up, eventually we get elliptically polarized lights. So from this is coming this characteristic of values we are obtaining from CD spectra, which is called molar ellipticity. So now, so what main data type we obtain from CD? Over there, you can see spectrum, which are coming from three types of secondary structures, like most common secondary structures that we can monitor in protein. So those are alpha helix, this in red, beta sheet in blue, and random coils in green. So these three kind of main types are information that we are getting. And basically, there are, so in sample, we of course do not have like protein completely made of alpha helices or beta sheets. We have some mixture of different secondary structures, which can interchange sometimes, and the composition can be changed depending on different conditions. And those changes we can monitor with circular dichroism. Basically, the first thing that you can see from your sample is what's protein folding, if it corresponds to the expected one. Also monitor different changes in composition of secondary structure, like how protein behaves under different temperatures, pH, or how small molecules can change secondary structure composition of your protein. And here, like, it is small example for lysozyme amyloid formation. So in this panel, we see different secondary structures of that amyloid under effect of inhibitors. So at the very right, under like high concentration of inhibitor, we see a formation just monomeric state of that lysozyme, similar to this one. So as you can see, just based on the effect of some small molecule, we can monitor those changes under different concentrations. So now closer to like limitations. So the question why we won't be using CD for everything, right? So it's really cool that we can get some structural understanding from CD, but it's not enough, right? So for CD, from a preparation perspective, there is some limitations that you cannot use everything in your sample. Let's say, so it's not good to use DMSO, imidazole for mammalites because they absorb light in far UV region, especially in this region. So you kind of lose a lot of data, and you wouldn't be able to get any understanding what you're getting from your spectra. And like for spectropolarimeters that I'm using at the molecular foundries, there's some throughput limitations since it uses in parallel just 6 cuvettes, and each cuvette is supposed to have 200 more than 200 microliters. So you can imagine that you use a lot of your sample, plus you're not able to screen in parallel like a lot of different conditions. And another thing that also is the most important in the limitation, and when SAXS goes in, is that differences in structures. So we can have very similar profiles of secondary structure content for very different molecules, and here this kind of creates a problem with interpretation of our results, because we cannot know for sure what type of structure we have in our solution. And the bridge between SAXS and CD, we can basically build this bridge since we have nice, good computational tools like FOX, BuildOMD. we can derive our structure from SAXS profiles and use its structure to derive CD profile. So basically, there is another computational tool called SAXS. So it takes PDB structure and converts it to CD profile. And from that CD profile, you can derive your secondary structure content and compare to your experimental one. So in this way, you can kind of validate your SAXS model applying also CD method. So here, I just want to quickly go over like CD workflows that I've been using here. For my project, I'm studying NSP8. So NSP8 is part of a replication transcription complex of coronaviruses. Today it was introduced by Mikko in the morning, and this NSP8 protein has this kind of golf club-like shape, but it's not always like this. So it was shown that it can participate in oligomerization, so it can basically form different type of structures like dimers, monomers, and it's not always that it maintains the same structure as you can see over here when it participates in complex formation for RTC. So here in my project, so what we wanted to do is to find out what the best salt conditions would be where we can get the most stable structure, right? So we would have this stable structure with potential application for crystallography. In sample preparation, as I mentioned before, we take 200 microliters of sample, as much as 0.5 milligram per milliliter of this small protein is enough to get a good signal. Sodium chloride is tolerable, but at very low concentrations. So in my analysis, I shifted to potassium chloride, which appeared to be better as it does not give very strong noise in this Paribus region. And the best was potassium fluoride, but as it appeared, we are getting some weird structures that not the same that we were expecting. So we stick with potassium chloride. And for buffer, we using a HIPIS buffer under 2 millimolar. As higher, it shows, as with higher HIPIS, it shows pretty increased interference with our data collection. And the best is, of course, phosphate buffers. But since we are trying to parallel our SAXS data collection, where we're using HIPIS buffer and a lot of data collected using HIPIS. So a lot of data collected from our collaborators with HIPIS, and we're using HIPIS in our CD workflow. And phosphate buffers are bad for SAXS, right? Yes. So yeah. Okay.
Just for SEC.
Oh, for ICC. Okay. I said for HTSAX.
For I triple two.
Yeah.
But
they couldn't like clock needle or. Now about data processing. Like we getting these not so nice for the first time curves. So these are spectrum. So they need some processing. So in CD, there are like several steps in data analysis. So I just quickly show some of those, like the main ones. First, we want to clean our CD spectrum by subtracting buffer. So we want to know that the signals that we are receiving not from buffer components, but solely from our protein. Next is data smoothing with some computational tools, and normalization means that we divide our raw values by concentration, number of residues in protein, and path length of the light. So we would have our spectra comparable to, through different experiments and samples. And what, like I mentioned before, some components can give us very significant noise. And you can see here is result for range of, so over here is range of potassium chloride concentrations in millimolar. In especially as a region, as a very far UV region, we see increase in voltage. So during like obtaining of data for CD, instrument maintains constant workflow, right? And to maintain this constancy, it optimizes HT voltage. So since it hits like particles that starts to absorb more light, the voltage goes up. And based on that differences in voltages, we can judge which region of our spectra is not good for analysis. And there's kind of a rule: we do not want to take data where we start and see saturation at 500 volts. Basically, for my analysis here, I cut data from starting from 200. So this region is optimal to analysis of beta alpha sheets, but I'm losing some data from that kind from flexible unordered regions. So, but still, I like having this region, I can say about secondary structure content regarding beta alpha and other secondary structures. And finally, we upload our spectra to best cell server. So it's Hungarian computational tool that allows you to obtain distribution of your secondary structures. I don't know if it's seen over there, but you kind of get a nice table with distribution of your secondary structures and with some statistical value which represent how is good fitting they did to your data. So basically, the server contains a lot of different references from different secondary structures, and it do regression fitting to your data, and based on that, it can generate percentage like close to what you would expect in your sample. Finally, some real data from HT SAXS and circular dichroism. So here in the left panel, I have NSP8, that proteins that I mentioned before, under different concentrations. So the first thing that you can notice is that molecular weight of protein itself is concentration dependent. As you can see, it increases from around 23 kilodalton to 45 with increase of protein concentration. And also it behaves differently along with increase of potassium chloride. At lower protein concentration, it tends to oligomerize. So basically, starting from 5 millimolar to 50 millimolar, we see linear increase in oligomers. So basically, we starting from monomeric state and going to dimers around 40, 42 kilodalton. And here to the right is secondary structure distributions from CD analysis. And you also can notice that within that region between 0 and 50 millimolar of potassium chloride, we observe this increase in alpha helices, so which allows us to think about that oligomerization is parallel with increasing alpha helices when we have formation of our dimers. Now we see that there is connection, this complementary connection between SAXS and CD data. And what about more atomistic structure relevant connection? So for that, as I mentioned before, we can use some computational tools. First of all, taking our profile from experimental data from SAXS, we can obtain like predicted model for that experimental profile. So here at low concentration of NSP8, we observed predominance of monomeric state. Taking the structure, we can convert the structure to CD profile. So over here, you can see a distribution of secondary structures from that model, which quite similar to the distribution of secondary structures from the experimental data coming from CD under zero potassium chloride. So as you can see that we have monomeric state at 0% of monomeric state of NSP8 at 0 millimolar potassium chloride. So now I'll talk about dynamic light scattering. So maybe a bit drastic transition, but this in other tools we can use for investigating what, like in my case, what type of oligomerization we can see, like if there is present any multi-state structures. So here, just quickly about how it works. So different particles with different size moves in solution, and once in DLS light hits those particles, they fluctuates, and that fluctuation directly is related to the size of that particle. So a bigger particle, it fluctuates slower, and with special functionality in DLS, we can do autocorrelation analysis, which eventually is related to that movements, that diffusion in of these particles in solution, and finally device gives us this peak distribution. Like, so these are beings that would say about distribution of different particles. Basically, we expect that each of those beings would correspond to different size of particles. And based on that, we can say about polydispersity. Like, in other words, if our solution, our sample is heterogeneous. Here I just showing you raw data which I obtained from DLS. So this is what we are getting from wired DLS device, so here at 0 millimolar potassium chloride, at the region between 1 and 10 nanometers, we see this one single peak around 3 nanometers. However, with the increase of the salt, like here at 50 millimolar, we start seeing this polydispersity. So there are more of those beings appearing. So you see, so here are 3 and more until we hit 800 millimolar, where that polydispersity decreases. So another thing is to consider in this experiment was buffer content. So this is just buffer alone at 100 millimolar, and we see some particle influence in the region, like from 100 to and higher. So this is something that could introduce artifacts in this region. So basically, if we see like two things very separate from each other, it means about polymodality. So it's not always meaning that you have like one small protein forming very big, huge things. So it might mean that some other components, like some salt, can form bigger particles, or some bubbles can interfere with your analysis, or you indeed have some big aggregates that just a bunch of unfolded protein aggregates and precipitate in your sample. So you need to be careful and always check this bigger region where probably we have some effect from other things coming there. And a good thing to assess your data in DLS is to compare your autocorrelation curves. So basically, this blue is your experimental data, so autocorrelated experimental data, and brownish. This one brownish is feed that DLS does. And as you can see here, we have some not the best feed, but for other data points, we see this good alignment of the brownish and blue curves. So this is one of the aspects to look at your data assessment during DLS. And finally, just to compare DLS and SAXS profile. So here we also see for molecular weight correlation with potassium chloride, and similarly that was shown before for particle size in DLS. And finally, the last methods that I've been using extensively, and I had a lot of different things going on with this and a lot of fun, is surface plasmon resonance. So this method is also kind of light-based. So basically, it allows you to get unlabeled kinetics of interactions of one-to-one molecule. So specifically, we have sensor chip with gold surface, and at that gold we have our protein or some nucleic acid attached. And once we flow analyte, like small molecule or another protein, and by attachment to the to that protein that attached to the sensor chip, we're getting changes in plasmon that's coming from activation by light. So basically, light activates electrons at the gold surface. Those electrons start to fluctuate, and we have this very sensitive environment of electrons that can change its fluctuation based on anything that float over that surface. So once we have interaction of, let's say, this red molecule attaches to this green antibody-like molecule, we get in this dip, and as much as dips formed, they transformed to this kind of exponential signal that is that's called sensogram. And here is example of ideal schematic sensogram. So you have association curves and a steady state where your molecule interact at the equilibrium, and then another exponential curve dissociation. So a bit about workflow that I've been doing here. So the first thing is to prepare your samples. Of course, you have like nice compartments inside of this thing. It looks small, but it's really huge. So we have this two sides where we have reagent plate, like 96 deep well plate, and over there we can also have 96 volt deep plate, but here it's just regular 96 volt plate. So these samples where we have things that we want to attach to the sensor chip, like your protein you analyzes or antibody, whatever. And so basically the first stage, so all of those are programmed in nice user-friendly software. So like they really have everything color-coded, so you always know what type of stage you are on. And so the first you have everything prepared. Then is the stage of capturing your molecule. So this another thing that terminology in SPR is quite weird, because things that we attach to the sensor are called ligands. But we used to know that ligands is something like usually small molecules that attaches to another protein, right? But here we can have anything like from like DNA, RNA, or antibodies, or any other proteins attached to the surface. So we attach this with different type of chemistry. So this is another thing that we can use different chips. So this is how the chip looks like. So this chip can have different polymers attached to that, and you can do different chemistry. You can attach your protein by just covalent coupling, but that means that you can damage your active site, right? Because covalent coupling. So the things that we are using in our lab is NTA-based attachment, where we have he-stacked protein and it attached by nickel NTA to the surface, or streptavidin-based chips we can attach biotinylated molecules as well. So you kind of precisely know where your label is, and you can attach it knowing that your binding site or any active site is safe. Then sample injection, we just flow analytes over the surface. We wash that any uninteracted, unbound ligands, not ligands, analytes, and then the last stage, which like might be optional sometimes, but usually you want to regenerate your surface. Like you can do complete regeneration when you get rid of everything, including protein attached to the sensor chip, or you can just get rid of any like analytes that were not washed off. So for that you can use like buffers with lower pH, with higher pH, with some detergents like SDS, or just high salt solutions. So like in our lab, we're using SPR Bruker from a Bruker company. So this SPR is called SPR 24 since we can use 24 samples simultaneously in parallel over 8 channels, and each channel can be used independently. So it's good that you sometimes do not want to use whole chip and paste it, right? So with this device, you can do different types of measurements, like from like regular kinetics to epitope characterization of your antibodies, quantification analytics to know like what concentration of particular analyte in your sample, as well as thermodynamics, as this device can regulate temperature, and but I forgot what's the maximum. Probably it can go to 60 or 80 degrees of Celsius. And finally, some experimental data that I obtained just recently. So being a part of Brave Task Force 5, we've been targeting viral proteins. And here what you can see very fast on and off kinetics with like pretty high concentration of analyte that just based on qualitative signal intensity, we can judge about this interaction. So basically here we're targeting macrodomain protein of VAV virus. So importantly here we have some molecules with higher signal, which corresponds to better binders like suramine and dasatinib, and some other like molecules with lower interactions, similar to signal coming from substrate ADP ribose, and no interaction whatsoever for one of our tested analytes. So this is just like example of fast obtaining data. So this can be done like within like 2 hours, and it's not used like whole capability of chip. It's just used some sensor spots, but not like whole chip. So you can imagine that you can have 24 of those analytes float over your surface, and you can do like 100 samples per day. So it's really good to obtain some understanding of interactions of your molecules. So what relations to SAXS? We intend to use NSP8, and it's like more or less stable form where we see like the one single monomer. So we intend to attach it to the surface and test how it interacts with itself when we have one protein attached to the surface and one float over it. And in this way, we want to see what the dynamics in terms of kinetics is for interactions between those monomeric proteins to form dimers. So like kind of in conclusions, I just show you some preliminary understanding on how potassium chloride affects oligomerization using 3 methods like SAXS, CD, and DLS, and some application of SPR in small molecule screening. And as a future direction, we want to test the NSP8 complex formation on SPR. And overall, as you can see, there are like several instruments that can be extended to for users' purposes as well at our beamline. And also, I would like to thank my research group, Sibbles, people from project of the DOE Task Force 5. Especially, I would like to thank Yang Chen, Todd, and Q for providing proteins. Thank you for all funding resources like again the Task Force 5, IDET, and especially thank you Molecular Foundry for providing training and maintaining devices that are located there like SPR and CD.